Thank you very much. Thank you for inviting me to come out and speak. So I'm wearing two hats today. Partly I'm the principal aquarium curator at the Hornbook Museum and Gardens in London, and also a co-founder of the Coral Spawning Lab. And myself and colleagues, we've been working on coral reproduction, but in closed systems, in land-based systems, for about 12 years now. And I'm going to kind of run through the potential of this and the work that we're doing to support both research and restoration programs around the world. First, so unfortunately, this is what many reefs are looking like around the world. This was a shot that I took in 2003 in Bonaire. This got hit really hard by the '98 bleaching event, lots of dynamite fishing out in the region, and all the structural complexity that the corals are building is obviously being taken down to very two-dimensional structures, and that obviously then has big impact on all the trophic levels that build from it. And so to understand if this can ever recover, reproduction is ultimately the way that seeding back into this degraded state may happen. So if there's a reproductively fit population that hasn't been impacted and its larval seeding can go back into this, then this can obviously recover naturally. More often than not, though, it needs a little helping hand, and this is obviously where reef restoration has sort of come into play. Understanding reproduction of corals, broadly, they're in two reproductive modes, or they express two reproductive modes. There's that of the brooders, about 15% of species are brooders, and they're characterized by internal fertilization and releasing of planular larvae. Now, it's often linked to the lunar cycle, so you get higher peaks of planulation near the full moon or other species around new moons. But they can spawn throughout the year, generally less larvae, but they're creating these peaks and troughs throughout the year. And we've been able to spawn brooding corals for many years in aquariums, Bacillivora damicornis, Tubastra kickinia, the bottom picture here, these are Fabia fragment from the Caribbean. But the main reproductive mode is that of broadcast spawners. So they're characterized typically by these sort of mass spawning events, releasing their gametes into the water, where fertilization is taking place externally. They can be either gonagoristic, so separate sexes, or more commonly hermaphrodites. So all the acroporids, for instance, are hermaphrodites. It's triggered by the lunar cycle. Well, progressive, I'll go into actually the parameters of this in a second. And then, you know, the main, or the first region that was ever studied and looked at this was the Great Barrier Reef, which, you know, is a mass spawning event, it's a synchronized event where the whole reef community is going over these few nights a year. So I kind of started learning about coral reproduction, going out on a Temporal workshop back in 2010. I've been working with coral husbandry my whole career for 25 years. And that really got me hooked about working with spawning. Unfortunately, I ran out of money to go on any more of those. So I went on this one workshop, came back to London, and was like, I really want to keep going with reproduction. So let's try and figure out what happens in the ocean and then replicate those in aquariums. And so started looking at the scientific literature, and we know that lots of papers have been published about some of the cues that trigger reproductive events in the ocean. So starting with things like species-specific drivers, so the age at which a coral becomes sexually mature. The thought a long time ago was that a coral needs to hit a certain size to proportion energy into reproduction. But then we know things like corals obviously have that symbiotic relationship with algae, get most of their energy from that. But they also are voracious predators. They eat a huge range of food types through heterotrophy, from bacteria right the way up to potentially even fish larvae and things like that. So once you hit the right size, they're feeding well, then the thought is that ultimately coming round in a clockwise pattern, there's a number of parameters that work at these progressively finer scales, which then trigger the coral to reproduce. So we have the seasonal temperature change, triggers the animal to start producing eggs. There's papers discussing regional wind fields, solar radiates, all the way through to tidal patterns and lunar cycles. And so I kind of looked at this and thought, well, I'll pick out certain parameters that would be needed to replicate that spawning. And so broadcast spawning events have happened in aquariums up to this point, but they've always been accidental events. No one had gone out and basically said, right, well, how do we trigger it and make it a predictable event? Once it's predictable, you get access to the gametes, and then it opens up a whole world of opportunity. So that's really what I kind of set the initial goal back in 2012. Could we put all of these parameters in place and have predictable spawning for the first time in captivity? And this is the Horniman Museum. We've got a small space behind the scenes, and we used some pretty rudimentary materials at the time to kind of put it together. And it worked. Within 8 months, we got the first spawning. So this was Acropora valida. I had lots of pieces of this same genotype in different aquariums. I pulled all those together because at the time it was thought that they had to hit a certain size. And so getting all these pieces together, sticking them on a piece of live rock, allowing them to fuse back together, I could build a big colony quickly. And then it would basically produce the energy to spawn. On the back of that, I then started my PhD 4 years later to write up all of these methodologies about how can we then spawn predictably. And we published this paper back in 2017. We wanted this to be open source, so anyone could pick up these techniques and then apply it to whatever research or restoration output they wanted. And so it describes how we program. There's a microprocessor attached to the system. And so as long as you can get data from the wild and from the location that the corals are coming from, you could create all this seasonality to get them to spawn. And so with this paper, we focus on Singaporean species and Australian, created temperature cycles, different photo periods and lunar cycles to trigger those events. Since then, we know the platform works really well. We've now spawned 50 species from every region, really, from the Caribbean to Red Sea, Indo-Pacific, Indian Ocean, into the Arabian Gulf. And really, the way I think about it is that land-based spawning and ex situ is the sort of broader umbrella. And then sitting underneath that gives sort of different areas of potential that we could use this as a tool. So whether it is understanding reproductive fundamentals, there's still so much we don't know about the way different species reproduce. From these reproductive events, how can we increase productivity so we create more corals for restoration? Thinking about the unique power that ex situ has over collecting gametes in situ, there's lots of interrogation work we can do. How do we scale up restoration outputs? And then also the role of land-based biobanks in closed systems to protect genetic material for species conservation. So I'm going to run through each one of these, apart from, I'm going to talk about the increasing productivity a little bit tomorrow. But really just thinking about reproductive fundamentals. I kind of use this species as an example. Acropora microcladus, it's a very common species on the Great Barrier Reef. There is literally no information about this species spawning. Bear in mind the Great Barrier Reef is probably the most studied area for reproduction in the world. And the reason behind that, it took me 4 years to figure out how this species reproduces. It's just because it goes really late after full moon, after sunset. So this spawns between 4 and 6 hours after sunset, as much later than most divers are in the water, because the main window for the GBR corals for Acropora is sitting between 2 and 3 hours. And so people just aren't in the water to observe this. So through ex situ, we're studying these or looking at these corals 365 days a year, gives you brilliant access to understanding of these reproductive behaviours. Starting to understand different grow-out phases. This species turns purple when it's young, and then that's its juvenile colouring, and then it takes about a year before it starts going into its adult colouring. So just some of these basic informations about the reproduction allows us to build on this. Let's keep this. Oh, video won't play. So where we have spawning events, we We've got the lab right next door. It means we can start applying some quite nice microscope imaging techniques. So I do a lot of fluorescent microscopy where I bolt on different fluorescent structures to understand the reproductive behavior. So at the top here, we've got an egg sperm package. So it's from an acropora and hermaphrodite, sperm in the middle, eggs around the outside. And by applying a fluorescent marker, I've marked up the DNA marked up the nucleus of the sperm. So these are actually sperm starting to come out of this egg sperm bundle during the breakdown. Bottom two pictures are during fertilization. So after 30 minutes after in vitro fertilization, each one of these dots again is a sperm cell. We start understanding where the entry site of the sperm is coming into the egg. And then this is a cross section 30 minutes after fertilization. The red is the egg. And then we've got the DNA of the sperm marked up there. And then the little red dots are the mitochondria at the back of the sperm. And then we use alpha tubulin to mark up the microtubules within the tails of the sperm. So you can start getting some really detailed understanding about the fertilization mechanisms of different species. We've then started taking that a step further and using different fluorescent markers to bolt onto structures during early settlement. So we can use non-toxic fluorescent markers that get incorporated into the calcium carbonate structure as the coral grows. So we get early life history information. So once they settle, we can apply these fluorescent markers and then you treat different cohorts. So we might look at different heterotrophic feeding and how that influences skeletal development. And you can use a technique called pushing and pulling. So we bolt on a fluorescent marker. It just gets laid down during that calcification. We then grow it for a little bit longer, bolt on another fluorescent marker, and it gives you two bands in the skeletal material. So it then starts helping develop your husbandry principle and how do you increase survivorship, health of the coral. So it's these types of techniques that we can use to interrogate those. Then I have lots of choices about how we want to manage those gametes that are released. So do we just let them all spawn in the tank and then you end up with multiple genotypes all spawning? It's much easier, but you're creating a mix of genetic material. So depending on the types of experiments we do, this is less staff is needed to manage a sort of an in-tank spawn. But the downside is it limits the type of experimental work we can do because we can't do pure pairwise crosses where we're understanding the maternal or paternal roots of inheritance. So it limits some of the type of science we can do. Whereas if you isolate each individual coral, once you start seeing setting, which is the egg sperm bundles coming up into the mouth, we'll then isolate them, bring them down to the lab, and then it opens up lots of other science that we can do to start understanding selective breeding for thermal resilience, understanding whether it's a maternal or paternal inherited trait. Those are the types of things we can start developing. The downside is we need more space. It's a bit more time consuming. There's more space required for the cultures to grow out. So there's pros and cons to each of these. The types of work that you can, though, do if you start doing pairwise crosses is you can start interrogating things like hybridization vigor, like I say, thermal stress type of work. In the Caribbean here, we've got two species of acropora. We have the pluriferas as a hybrid. But out in Australia or Indonesia or wherever that might be, there's hundreds and hundreds of acroporas. We know that hybridization happens on the reef. We know that hybridization vigor as well provides increased thermal resilience. So start trying to interrogate where the boundaries are between which species can hybridize and which can't within the group. And so we've done maybe 80 different hybridization crosses. Generally, there's something called pre-zygotic barriers. Most of those crosses don't work. But it's really interesting when you start understanding which ones can and which ones can't, and then it gives you the material to then take that step further and then do thermal stress work. So this is just an example with Acropora and Meliopora. We can fertilize Meliopora. For some reason, Meliopora seems very receptive to taking sperm from lots of different other species, whereas it often doesn't work the other way around. But you really need to be doing that isolated gamete collection to make sure that the sperm isn't in the water before you start doing these. Thinking around other species, so this is not necessarily applicable directly to restoration, but we've started doing a lot of work with, in the aquarium trade, it's called LPS, large polyp stony corals. Now, for the aquarium trade, these can't be asexually fragmented like acroporids, or not. They can, but not commercially. So the supply of this into the aquarium trade is fully extracted. So each individual polyp, this is one polyp, so they only form one polyp, they're collected and taken into the aquarium trade. So you've lost that genetic material on the reef. These are incredibly high value. If they're the right colour variety, they can go up to $6,500 for one individual coral. So there's a high demand on this for the aquarium trade. I started wanting to look at how do we reproduce this? How can we flip that model on its head, work with reproduction, and actually start poultrying specimens to go into the aquarium trade, removes that need to harvest from the wild, and looking at alternative sustainable livelihoods. So this was actually all done in my kitchen. I've got a spawning system in my kitchen to teach my boys. We've done 16 species now. This is the first time this has been done in the world. We stayed up all night and preserved all the eggs and ran the scanning electron microscopy to sort of document the embryological development of the species. And it was always thought that this would be a really slow-growing species. Is this an individual polyp that might be 10 years old? But we actually don't think that is true at all. We're getting really fast growth rates, and so it opens up this aquaculture potential. We've done a similar thing with Capitalophilia. Again, this is the first time it's been done. These are now 2.5 years old, very easy to get a marketable size within 18 months, really. So it opens up lots of opportunity to culture those in land-based facilities. We want to take that a step further, though. So we won a Cordat grant, and we're going to start exploring this idea in Indonesia. So we're building the systems in the UK at the moment. They're going to be going out to Bandarwangi in eastern Java. And rather than taking these types of species from wild harvest, we'll start looking at culturing these for the aquarium trade. Now, we know that the colour varieties have the highest demand, but those that don't have necessarily the colour that have that high demand, that becomes the restoration material. So you can bring an income stream into a restoration model where you're funding it through the aquarium trade and then actually paying for that restoration. So we're really keen on looking at these circular economic models and funding restoration in a different way. And so this is a work that we're going to be doing with the Indonesian government, with a view that this will then create the model that will roll out across the region. And again, we'll be working with these LPS species. So ex situ spawning also provides a unique potential that you simply just can't do in situ. And that's that we can phase shift at multiple temporal scales. So all of our spawning happens during the day rather than at night. And we've designed the systems where we actually make sunset happen about 10:30 in the morning. And it means that the spawning then starts happening. If we're working with Great Barrier Reef species, for instance, Acropora tenuis normally spawns between well, it's actually proper Kentai now, but from 3 to 5 nights after the full moon, generally goes about 1 hour after sunset. So if we make our sunset happen at 10:30 in the morning, we then have spawning at 11:30. We then can do all the fertilization, have a lunch and a tea break because we're English, and then it kind of picks up about 11 o'clock that we have media flora and all the other species sort of fitting in. We've then taken that a step further where we move the lunar cycle. So if we look at our staffing and we don't have enough staff at weekend, we bring our lunar cycle in line, so we move the spawn so it happens within our working week. And then we've also started then looking at how do we phase shift at different times of the year. And the whole idea of this, I stumbled across this actually purely by accident. We were initially working with Singaporean corals. We spawned those in March and April, and then needed to run an experiment where I had to reprogram my systems to follow an Australian spawn. So I kind of pushed all the Singaporean corals up at the back, we brought in some Australian corals, we reprogrammed the system, and we actually found that the Singaporean corals then jumped onto the Australian cycle. So they'd already spawned in March and April, and then they spawned again in November, December. And we're like, hang on a minute, we've got a brilliant opportunity here. to start interrogating both inter and intra-specific hybridization, creating different geographical crosses. We're not advocating this as something that should happen for restoration, but it's understanding the tools of the potential of this. So we ended up taking a proper hyacinthus from Singapore, we had hyacinthus from Australia, and then also Mediapora. So we made intergeographical crosses both within species and out of species. And then we ran a thermal stress experiment with both the adults and the juveniles. And the idea being that Singapore is much warmer water, so within their genetic code is the ability to evolve and cope with those warmer waters compared to Australia. And then after that thermal stress, we ran metabolomic analysis. We actually found that the juveniles are really plastic. And this is really important from a restoration potential, that actually the juveniles have this great adaptive potential where the adults just simply don't. So all of those Australian corals that put into the thermal stress bleach very quickly, die much quicker, because it's just they've not been put on the treadmill because it's not the region they're working with. Whereas the juveniles have that adaptive potential. So it's making sure that within that early stages, giving that thermal stress and sort of prepping them for the future. We published one paper, we're just writing up the plasticity of the juveniles at the moment. Then we started taking that a step further and think, well, we know we can move them, so how easy is it to move? So this was a study that we ran over 3 years to understand which of the parameters we would need to move. And so we had 3 different systems in this scenario. We have one that was kept in sync with Australia. We were using Galaxia fascicularis. And then we phase shifted another one by 4 months and phase shifted another one by 8 months. So this is looking at a circular model, 0 being new moon. As you travel anti-clockwise, you're moving through the year. And so ultimately, this is really important when we think about access to the gametes during natural spawning events is really limited. We've got a few nights a year to collect the material to do the settlement. So if we can start accelerating that and getting more spawns a year, it then opens up lots of potential for restoration output, for research, whatever that might be. But understanding what are those parameters to move was the key. That kind of then led on to actually the formulation of the coral spawning lab. So Cal State University had asked us to build systems for them. They're doing a lot of work with CRISPR-Cas9, so injecting eggs and doing a lot of gene editing work to understand how do you develop that as a tool for restoration in the future. So by injecting the egg, you're either doing things like gene knockdown therapy or gene transplants. Access to the eggs is really important for that because it's a difficult technique. So we designed a system that ultimately would hopefully give them 24 spawns a year, using 2 different species that naturally sit 1 month apart. So we created 12 spawns, or 12 spawning windows, but then with the natural 1 month delay would give 24 spawns. The first year actually gave us something completely different. We had 95 spawning events. Out of that, they yield 30 of those spawning events had enough to do their CRISPR-Cas9. So it is really accelerated the type of platform to interrogate these types of tools and development. And this is Max is doing his PhD, and this is some of his data collected from those. So like I say, that formed the coral spawning lab. Mike and myself were getting asked to sort of help with lots of systems around the world. So we formed the coral spawning lab initially with Vince, but Vince isn't part of that anymore. The 2 of us are now moving forward. And we have these off-the-shelf systems which are designed off the original work of the PhD, and then we've sort of taken those a step further and start containerizing them so you can put multiple systems in a container, phase shift to create more access to material as time goes on. This started literally, and then COVID kicked in, so we thought maybe 1 or 2 systems might go out, and it's gone mental. So we've had 70 systems go around the world, and they're really the platform that people can then use, whether that is for research purposes. So a lot of the work in Middle America is looking at genetics work, or more commonly for restoration, so Coral Vita, Mo Marine Labs, work out in Sonava and Six Senses. These have all been used for restoration tools. So it's a platform that people can use it for whatever they want to use, really. So we've got the spawning. What do we then do with it? I highlight work I've been involved with James Guest and Adriana Humanas at Newcastle University. They've been working in Palau since 2018. We started out there, and it's looking at this question about, is human-assisted evolution a thing? So this was the whole question about, can you thermally or breed thermally resilient corals? The paper that Adriana published here looks at how long do you need to keep these animals in a land-based facility before you plant out. And so in this paper, you see all of these corals at the top here are the same age. They're 2.5 years old. In D, this was kept in a land-based facility for 5 months. E is kept in a land-based facility for 11 months prior to outplanting, and F is where we've kept it in a land-based facility for 13 months, then it's gone into an ocean-based frame like this, and the mesh is big enough to allow parrotfish to get in, small parrotfish, but excludes the big parrotfish. And I think this would be a really similar example in somewhere like Bonaire. It's very true of the region in Palau, which has a very high parrotfish grazing population. And what we find is that if you plant them out too small, they're just getting hammered the whole time and they can't get a foothold. Whereas, the ones that are kept in a land-based facility, then going into an in-situ nursery, these are then spawning at 3 years old and contributing back into the repopulation. So just understanding that, again, it sort of can fit into a restoration strategy about how best to use that reproductive material coming out. We've then sort of taken this a step further to see if we can create much larger shipping container designs with solar panels to take it partially off grid. And then we've got the spawning. How do we then maximize the efficiency of those spat that are produced from these reproductive events? So this is a spawning that I had in my kitchen. I kind of want to highlight, if I could do this in my kitchen, it definitely could be done in lots of places on a much bigger scale. But I work with Acropora muricata from Indonesia. I've got a couple of genotypes of that, and it spawns really regularly for me. And I kind of reared just an eighth of that material, settle these onto 64 plugs. And rather than treating this one individual plug as a recruit, basically after 2.5 months, you see multiple spat on there, average is about 5. And then spent the effort to cut each one of these out. So from 64 plugs, there was 618 recruits, could then actually cut out 412 of those, and that's led on to 364 individuals that are 11 months old. So by putting intervention at this stage, we maximize the genetic potential coming from these reproductive events. We then need to start thinking about how do we maximize the space efficiency in land-based facilities? So we started thinking about how do we make an energy efficient system taking inspiration from nature. So the idea of this, our V-shaped raceway, is we're taking inspiration from a river system that meanders backwards and forwards. And ultimately what we're doing is pumping, the filtration is just below here. All we have to do is pump from this end section to this end section, and then it slowly rotates and meanders backwards and forwards so that you're actually using a low energy watt pump to move water quite efficiently. These are modular, so the idea they can be expanded to whatever space that you have. So each one has a 2-meter V-shaped raceway. On these 2 meters, we have these these maternity trays. We can take 216 plugs. So each section of V-shaped raceway can hold 2,000 plugs. And then here we've got 4 sections. We've got over 8,000 plugs in this system. We then try to expand this out. So when you start then bolting on different sections and making this long meandering river, all we're doing is pumping from here to here, but then allowing that natural meander to come backwards and forwards. So we reduce the energy demand, move the water very efficiently. And the V-shaped design allows sedimentation to fall to the bottom, reduces that sedimentation on the spat, which we know inhibits sort of survivorship. 2 of these we'll put then out in the moldies. So it gives a land-based facility with way over 60,000 land-based spat can be grown out prior to that outplanting. We also put a thermal stress system in here so you can start ranking your individuals so you know which individual you should be spawning from to create stronger corals. And then just thinking about this horrendous picture around the world, we know that reefs all around the world are getting absolutely hammered, and the role that biobanks and maintaining some of this genetic material in living biobanks is now becoming, unfortunately, an increasingly important tool around the world. So the Coral Conservatoire, based out of Monaco, it was founded a few years ago to bring genetic material in. Obviously, in America, there's the Florida Reef Track project with all NOAA or AZA institution here is Florida Aquariums, but much of that genetic material from Florida Reef Track is now swimming in lots of different aquariums around the US. Similar story is happening in Australia, and there's discussions about a new facility being built in Qatar. And this is, unfortunately, last-ditch efforts really to make sure that we bring individuals in before we lose them. So just in summary, we developed land-based systems that provide access throughout the year to really study reproductive behaviours. I think there's amazing opportunities for different circular economic models that brings industry into play in restoration outputs. I'm going to touch a little bit more on this tomorrow about other programs that we're working on. Phase shifting gives a lot more access to material. We can do that at lots of different temporal scales. Husbandry bottlenecks, again, I'm going to talk a little bit more about that about tomorrow. How do we then increase more productivity from these events? And unfortunately, biobanking is part of that solution to ensure species survival in the future. Loads of industry and partners have supported the work over the last 12 years that I need to thank. It really wouldn't be possible without any of them. And from my Acropora Pora embryo, thank you very much for listening. If there's any questions, thank you.
That was very impressive to learn more from me, completely new, and I think especially as well for us in conservation, we have a lot of in situ work going on, and it's nice to learn as well all the potentials it's possible with ex situ work. I'm first going to open up the floor for some questions for the people present here, in the meeting on Bonaire, and then if there's time left, we'll open up for people in Zoom as well. I see a first question from Karen, and Karen, I want you to ask to come a bit close to this one. The people in Zoom can hear it. Yeah.
Okay, so it seems like your process are mostly focused at thermal resistance. And I know it's hard to test, but do you do anything with resistance?
We have not done it. Well, I should say that Professor Mike Sweet was one of the co-founders. That is his, he's an expert in coral disease. We haven't started doing that yet, looking at disease resistance. But absolutely, it needs to happen, for sure, with the situation in the Caribbean. Is it possible to create disease-resistant corals? We have not, no. Mike does a lot of work with probiotics and that. So we do a lot of probiotic work and looking at early post-settlement survivorship. So kind of work on boosting the health rather than subverting the disease.
Thank you for your presentation. Really great work. I was wondering with the LPS selection, like these are for the aquarium trade and these are for restoration. Is there a genetic basis for the color variations, and are you not concerned that you're sort of selecting for a wild genotype that is different than the?
That is a really important question, and one that actually, so the selection pressure of collecting them from the wild is specifically going out to collect those colour varieties that are bright and more fancy. We have absolutely no clue about what ecological significance that has. Do those colours play any ecological role or not, or is it just a byproduct of that? So flipping that the other way around, everything that's dull is left on the reef, and is that having an ecological problem? In terms of the mechanisms, I haven't put any slides in this, but we've been doing a lot of work thinking about colour, and inheritance of different colours and the mechanisms of creating chimeras and stuff like that. I can't answer that from the point of view, does that have an ecological problem by putting those back? I would say it's the opposite way around, the fact that those colour varieties are being pulled out of the ocean and we don't know what ecological significance that has.
After reading ex situ with manipulated systems, that they're basically off track with the natural system, what does it take to readjust the offspring to get them to go back to the natural system?
So, I mean, certainly we've been doing a lot of work thinking about what additional, and I'm putting some slides in this tomorrow, things like different heterotrophy, how we can boost resilience through heterotrophy to make that transition back to the ocean as easy as possible. Certainly, so actually while I'm here, I'm taking light reading data as well. So, you know, the technology that's available from a lighting point of view, there's lots we can do to photo acclimate them prior to that. So those are kind of the considerations and how do we boost them up? How do we create that initial thermal resilience and hope that sticks? How do you photo acclimate them prior to going out? And the example in Palau is exactly that kind of process as well. You know, I suppose every system is going to be different in terms of how long should it stay land-based, whether there should be an intermediary in situ base or directly planted out. So, does that answer?
Thank you. The fragments that you were testing for heat tolerance, you said that they're more resilient compared to the adults.
Mm-hmm.
Do you know that if the resilience is also genetically based?
So the data on that, is showing that there is thermal resilience that is genetically based. What I'm going to present tomorrow is also showing there is an environmental base to it as well. So I think we need to work on both of those routes. So James and Adriana, they basically had 400 corals tagged on the reef from 4 species. Pieces were taken from all of those and thermally ranked. So they knew which ones had thermal resilience from the broodstock. And that's what created the pairwise crosses with that. So we created a high-high cross, a high-low cross, a low-high cross, and a low-low cross. And that allows you to then interrogate whether it's maternal or paternal route of inheritance. There's a paper that we're hoping is going to get through the next phase of this, but basically they've shown that there's multi-generational inheritance. So not only does it pass from F1, we're also seeing that sticks into F2, which is really encouraging. Yeah.
Thank you very much, Jamie. I think we can open up for one question from the Zoom chat.
Yeah, we have a question from Diana. She is working in a European project for Reef Restoration in the Mediterranean.
Yeah.
And she is wondering whether you have worked with soft rocks.
Yeah, so at the museum, we've got a PhD student looking at unicellular ventricosa. So we've modified a system specifically for unicellular. The geographical range is from the bottom of the Irish Sea right the way into the Mediterranean, so it will be a species that Diane's working with. We have been getting excellent growth rates, so we designed the system to create different tidal patterns. It goes backwards and forwards, and then we feed on the making tide, because with gorgonians, basically the tidal pattern basically bends the polyp. That creates back eddies, which really drives the feeding efficiency. So we've been working with the biology of that. In situ growth rate, 1 centimetre a year documented. We hit 3.5 centimetres a year, so we've really ramped up that growth. The plan is we're trying to look at reproduction of that. So if all going well, August, September this year, we'll hopefully have that first spawning. And Lisbon Aquarium are doing some brilliant work with this as well. So she wants to reach out to Nerea at Lisbon.
Great. We have a lot of great questions in the chat. So, but I told them that they can reach out to you through us. We'll make sure that everyone gets their answers.
Thank you.
Yeah, thank you so much.